So Dr. Strain completed his MD and PhD actually here at UCSD and did some postdoctoral work in Doug Richmond's lab and is now an assistant professor of pathology. And today he's going to tell us about a very interesting project entitled Open Source Digital PCR of HIV RNA and HIV DNA.
So I'd like to tell you today about, shift gears a little bit, talk about a different type of project. And the basic goal of this project is to develop an open source platform for digital PCR generally. And the application that we're most interested in is HIV RNA and perhaps also DNA testing. So there is a tremendous unmet need for nucleic acid diagnostics and particularly for HIV viral load testing. The World Health Organization recommends that all patients be tested before and during treatment with antiretroviral therapy, and even that is only a small portion of the need. UNAIDS has set the quite lofty goal of what they call 90-90-90, that 90% of all patients living with HIV know that they are positive, that 90% receive sustained antiretroviral therapy, and that 90% of those are suppressed. If that were the case, the total demand for antiretroviral testing or for viral load testing would be upwards of 50 million tests per year, which is well above, almost a 30-fold higher than what is currently achieved. As recently as 2013, an Médecins Sans Frontières study found that fewer than 1% of patients in sub-Saharan Africa currently receiving antiretroviral therapy had ever been tested for viral load. So the gap between the goal and the current situation is indeed tremendous. And the main challenge here is delivering these tests, which are available in the developed world at a cost that is feasible in the developing world. The current cost as available in limited resource settings varies between $10 or $9.40 actually is the price that Roche is currently offering. In many settings, up to $72. And the Gates Foundation and other organizations have suggested a goal of $2 per test. But I would like to mention that all the focus has generally been on the cost of disposables and reagents per test, which is, of course, critical. There are also very significant instrument costs that are often not completely accounted for. So the four commercially instruments that are most widely used, together with a biocentric platform, which is not FDA approved but is being used some in other countries, are shown here in this plot, and you see basically none of them cost less than $50,000. The accounting of this is sometimes swept under the rug. For instance, even in a recent MSF Access report, they concluded that only 3% of the current cost of antiretroviral testing in limited resource settings was due to equipment. However, to achieve that, based on their cost numbers and these equipment costs, each instrument would need to do at least 600,000 viral load tests within its lifetime. To say the least, that would require excellent preventive maintenance, which is generally not available in limited resource settings. Additional challenges that remain to be met include cold chain storage and delivery of reagents in some locations, particularly in the most turbulent and violent areas. So our approach is actually quite different from what has been done in the past. So what is primarily being done at present is there are efforts to take the FDA-approved tests that are used in the US and to deliver them throughout the world. However, those instruments and technologies were developed with the goal of making money for the manufacturers, in part, and so there are lots of costs built into the equipment and the process itself, but in terms of intellectual property and additional costs that need not be removed. So again, looking at these equipment costs, these $50,000 instruments are made up of usually a few $100s worth of components. All the rest is value added. So our approach is to make a modular design with all open source components, and that the term open source comes from software. It's now used extensively in hardware design, particularly in connection with things like the Arduino platform, because almost any piece of computer hardware has internal microcontrollers that have their own software. So that is open as well. The overall extraction protocol, the overall assay protocol that we're looking at here, the first step, extract nucleic acids from blood. We're using a magnetic bead technology. There are a number of these available. Then the quantification step is achieved by first partitioning the nucleic acids into a number of, actually thousands of different microwells in a plastic chip, and then performing PCR using a custom-made open source thermal cycler. Then counting the positive reactions by low resolution fluorescence microscopy. And finally, to convert that into a number, there is a software component that analyzes those images and performs statistical analysis. The critical piece of this that we achieved first that made it possible to produce the chips that we need to do this testing was to develop a new process for making these plastic microwell chips. We and others had previously tried to do the same thing using different materials. Typically in the micro device development process, people start out using something like PDMS, which is very popular for academic studies, but not really scalable for industrial applications. So we here have developed what we're calling a soft lithography process. This was done in collaboration with Alex Groisman's lab in the Department of Physics. The process begins with standard lithography, the technology used to make most small computer chips, which uses UV light to produce a pattern on a silicon wafer. That is then used to produce an intermediate in PDMS or silicone, a soft material. We then produce another intermediate, an epoxy embossing tool, and then use that finally to basically stamp out the pattern in plastics. This soft embossing process, as I said, it was critical to our development because although making plastics is very cheap on a very large scale, it actually is not all that cheap on a small scale, and particularly when you need quite high resolution. And so if we had done this using standard metal tools for embossing, it would have cost probably $25,000 or so for each chip design. And of course, in the early phases of the design process, you go through designs pretty rapidly. So the cost of development and prototyping would be prohibitive by previously existing methods. We've also developed an open source thermal cycler, which was designed specifically for cycling these small chips. Because of the small dimensions, primarily it's able to cycle somewhat faster than most of the less expensive commercially available thermal cyclers. At present, it takes about 30 minutes for 40 cycles. We can probably shorten that if we need to. It's not the rate limiting step at this point. This is controlled by an Arduino microcontroller using an open source firmware. There's a large community that has developed around the world that is using these to automate all kinds of things. And it's our hope that that community can be called on in the future to help with repairs and replacements, as well as just in improving our design. So we're already on the second version of this prototype. The first version had one component that cost $300, which we decided was a bit too expensive. So the version 2 now has brought down the total component cost to just under $200. So the images produced here, this gives you an example of one of the images that comes out from this process. And so there's a red loading dot here to indicate where the wells are, and then the green, which is FAM-Zen detecting HIV gag, shows you the positive wells. As you can see, there's quite a bit of dirt on the chip. So you can see, you can probably make out there some very tiny little dots of green that are not in the rectangular pattern of the wells. That's dirt, fluorescent dirt sitting on the top of the chip. So a critical piece of the software image analysis is to eliminate those artifacts. And once you do that, the wells stand out quite clearly. Dim background allows you to calculate how many wells there are, which is important for determining the denominator. And the bright fluorescence identifies the positive wells. So this produces a signal that is similar to the digital PCR, to droplet digital PCR, other technologies, but also gives quite a bit additional information. So shown here that for each individual well, you get not a single, you get, of course, an image, right? So you can see that there is spatial structure within there, and the patterns vary considerably. So this is one of the elements of the protocol that's still under development, but it's already clear that this spatial information can be used to eliminate a substantial number of the false positive events, and it is my hope that we'll be able to get rid of all of them, which would move this beyond even the droplet digital PCR systems that are currently commercially available for $100,000. The big advantage of doing this all with PCR rather than one of the isothermal technologies that is popular now for in-point of care diagnostics is that there is a tremendous amount of literature, tremendous knowledge of the use of PCR. So the molecular biology all transfers over really pretty much trivially. And in particular, the primer and probe set that we are using here, uses HIV GAG, was originally developed by Roche with their first version Amplicor assay, and then we, in studies I've shown here, had previously demonstrated that by droplet digital PCR, this primer probe set could be used to measure viral loads with an accuracy that was comparable to, actually slightly better. The coefficient of variation here is slightly better than what Abbott reports for their own assay. We compared with Abbott and got quite similar results. So we knew going in that the molecular biology worked. Although I should mention that in order to make this work in the chip system, it was critical to use the double quenched Zen probes, which are currently exclusively available from IDT. One of the goals of the wet lab portion of this open source project is to try to make sure that we are not dependent on any one manufacturer for any one component. So at present, that it has not been achieved. However, it is true that IDT at least makes those reagents available to everyone, anyone with no licensing costs, in contrast to probes available from other manufacturers. At this point, I can tell you that the assay performance is not significantly different, no more than at most twofold less sensitive for HIV DNA detection than what we've previously published for droplet digital PCR. So that corresponds to 10 copies per million cells. And that is actually substantially better than the same assay implemented with the only commercial equivalent to our system. So LifeTech does make a system that they've released in the last year and a half or so that uses a plastic microwell-based chip about the same size. Instrument costs $45,000, and it is plagued by, based on the one publication available now from those data, by much higher background levels. So we are substantially better than that. In HIV RNA, we're currently in the process of doing analysis with a blinded panel of samples. So since I'm still blinded to those data, I can't tell you exactly where we are, but I can't except to say that the lower limit of quantification is currently, based on analysis of separate negative samples, about 60 copies per mil due to occasional false positives. But as I mentioned, I do think it will be possible to reduce that primarily through better image analysis. So future work, we are doing additional validation to establish the operating characteristics of our complete platform. And then we are interested in looking at additional targets, and I'm very open to other suggestions. If anyone out there has ideas for other nucleic acid diagnostics, it could be useful in limited resource settings, particularly quantitative diagnostics. That's something we're interested in. There's ongoing development to improve various aspects of the engineering. The main thing is we like to integrate all the different components and have it be as user-friendly as possible so that it can be used by people with minimal training. Also developing a chip to do the HIV RNA extraction within an enclosed chip to make that less dependent on technician skill. And then we will be field testing this in Hillcrest and taking patients who currently come in for the early test. These are patients who believe they may have contracted HIV in the past couple of weeks, some very high-risk patients. And at present, those who are HIV antibody negative are tested for HIV nucleic acids with the hologic GenProbe test, which is sent out. It takes about 4 or 5 days, so we'll be doing those testing. We'll test those patients in parallel the same day in order to get the more rapid turnaround and validate our test at the same time. And then, of course, the real goal is to test this in limited resource settings. And the site that we're most interested in starting out with is Maputo, Mozambique. But again, if any of you out there have other ideas, I would be happy to discuss additional possibilities for deployment and future testing. This project, as I think is probably apparent, was a highly interdisciplinary project that could not have been done without collaboration in the School of Engineering, School of Medicine, and School of Arts and Sciences, yes, the Physics Department. So thanks to all of my collaborators, and in particular to Bill and Caitlin, who have, as shown here, run back and forth between all these different places to coordinate things. Thanks.
Okay, a couple of short questions.
Hi, thank you for giving this talk. I just had a quick question about, you mentioned an interest in doing work in Maputo. I was wondering if you know about the stability in like high humidity or high heat environments for the device?
Well, so the device itself, that should not be a problem. We've done some testing of that already, but, well, actually, I did run the original thermal cycle prototype in the incubator. So the biggest challenge is the reagents. So one of the things that we would like to do actually is to move to completely lyophilized reagents so that the reagents can be stored above room temperature and not require cold chain. But the instrument itself, that is an issue that we're very interested in, concerned about, and I can tell you the thermal cycler is fine, and I believe all the other components are. But we will definitely test that before deploying.
Thanks.
There.
I actually have two questions. One is regarding the throughput. How many patients can you put in a single chip, counting for, I don't know, replicates and different dilutions and so on? And the second question, so you talked about it as based on blood samples, but can it be done using other bodily fluids, saliva, for example? Yes, so certainly other fluids could be used. We have not yet tested dried blood spots, but there is now an extensive literature on doing extraction from dried blood spots, so I expect that would work, but that is something that we're interested in. And the way it has been designed, in answer to your first question, is just basically one sample per chip. So they're small chips, they're 1 by 1 centimeter chips. The current design of the cycler can accommodate 3 of them at once, so the idea was that we would make many of these instruments, but they can also be made larger if someone wants to process on a larger scale. So this was designed more as something that, could be even a point of care instrument. In a large center that's doing large volume processing, there would be an array of them.